We are so excited. People are joining us today. So we're going to let the attendees come in since we just started our webinar, and then we will get going on our awesome live event live event today. So welcome everybody. While we're waiting before we get started, my name is Sarah Ecker. I work at the National Science Foundation. We'd love to hear where you're tuning in from. So if you can just put in the chat, where are you tuning in from, and maybe how many of you there are. Are you a classroom? Is it you and your family, you and your dog? We'd love to hear where folks are from today. So I say Colorado. Oh, hello, 4th grade class. Fantastic. Lots of Virginia, Nebraska. Oh, I love seeing people from all over the country. So I'll say good morning or good afternoon, depending on where you are. Time zones do that to us. Kentucky. Awesome. Well, welcome everybody. We're gonna leave that chat open for folks to keep putting in where they're from. But I'm gonna just quickly get started. As I said, my name is Sarah Eckert, and I am a communication specialist with the Office of Polar Programs at the National Science Foundation. And I'm so excited to welcome you all to our live event with some really, really fascinating researchers who like to study worms and other invertebrates. So before we get started, just some housekeeping pieces. All of our attendees are muted and your videos are turned off. If you have any questions, please use the Q&A function. Virginia and Holly and the rest of our research team will use that, look through those questions and be able to answer them. Also, this webinar is being recorded. So after the event, I will be sending out a link in the next few days. And yeah, so the webinar is approximately an hour, and we understand classes sometimes are shorter. So we will be sending that link to everybody after. Awesome. So welcome. We're doing a live from the Nathaniel B. Palmer, kind of. I'm going to let Virginia give you an overview once we get started. But because of our current research situation and unfortunately COVID, we have some folks on the Nathaniel B. Palmer and also in their hotel rooms. So I know they're going to talk a little bit, give you some information about who is where. But the Nathaniel B. Palmer is one of our research vessels that explores the waters of Antarctica. They're headed out on a cruise, but currently the Palmer is in New Zealand at port. So today we have joining us Holly, Bick, and Virginia Shute. They are both fantastic researchers and storytellers who are going to be leading us today. And joining them is a team of researchers, grad students who are on their way with Holly and her team to join this research vessel. We also have some folks on the Nathaniel B. Palmer who will be giving us a virtual tour, and just so you know who they are as well. So with that, I'm going to have our stop sharing the screen and turn it over to Virginia.
Good morning, everyone from New Zealand. It is 6:05 in the morning. That's how excited we are to tell y'all about what we are going to be doing for the next couple of months. Before we get started, I want to echo what Sarah said. We are in 2 groups today. Group number 1 that you will meet are researcher scientists. They came here from their universities in the US, and what they're doing in Antarctica is just a part of their job and their research program. Group number 2 that you will meet are the science crew that's on the Nathaniel B. Palmer right now. If you can see from my background, I'm in a hotel. The scientists, researchers, the research scientists are in a hotel while the science crew is on the ship. We are staying separate right now so that we can all stay safe from COVID. So I'm going to have us meet the scientists first. These are the team members in the hotel, the ones who this whole thing is just part of their job. We're going to tell you who we are, and we're also going to tell you a quick story about something gooey or squishy or generally kind of like blecky that's happened to us in our jobs. I will go first. Hello, I am Virginia Schutte. I am a marine biologist who makes stuff for the internet that connects people with science. On this Antarctic expedition, that means organizing stuff like this discussion so that you can come along and see what life is like when you're a scientist. When I think of gooey, I remember when I was doing my own research in the tropics, I was snorkeling in forests that are right at the edge of the ocean. So the trees will grow their roots and their branches down into the water. And those roots and branches will get covered in stuff like corals and oysters. It's kind of like swimming through a coral reef instead of being on top and looking down. So I was snugged up against a root, touching it, doing stuff, measuring, when I felt something go from my hair down the back of my head. And I mean, that happens all the time. I get dripped on. There's like little leaves and stuff that drop down. But this thing, instead of plopping down in the water, it crawled around and started to go into my ear. And I tell you, my hand has never moved so fast to dig that thing right back out. It did not lodge, it did not stay. But I remember taking a really good long break and like thinking about my life decisions that brought me to that point. And I was able to get back in the water, but not a black experience I would like to repeat. I am going to pass the baton to Holly. Will you please introduce yourself?
Yes. Hi, everyone. I'm Holly Bick. I am currently an assistant professor at the University of Georgia. So I'm research faculty there. And I'm leading a team on this expedition to Antarctica. And so we have a couple of my team members here. But I study nematode worms, roundworms. Yeah, but I love all types of worms generally. So my gooey story is probably about the worm that the type of thing that I do not like. So I always joke that the reason why I love the ocean and I work in the ocean is basically because I've had too many traumatic experiences with freshwater. So I grew up with a river in the back of my house. I used to go to summer camps in Massachusetts and New Hampshire, growing up in New England. And I have way too many memories of like leeches on my legs, like when I was swimming in lakes or, like fishing in rivers and everything. And so I just like, leeches are my nemesis and literally like my mortal enemy when it comes to gooey things. And they're pretty cool because they have anticoagulants in their blood. So I respect them, but I don't trust them. And that's why I work in the ocean on different worms.
I get that and think that's a really nice choice. Thank you, Holly. Next, we will have Miriana. Can you please introduce yourself?
Hi, good morning. My name is Mirayana. I'm one of the Holly Beck's team, so I'm her student in the University of Georgia. I'm here as a scientist, and I'm very excited to find my worms that I love here in Antarctica. So one story that I have many gooey stories about worms because I have been working with these worms has been like 20 years. So I have a lot of preparation of those worms. So one that I remember is not about the worms, but about a situation where I had a polychaete living on my hair for a little bit. It's where I was processing the samples that we got from the ocean. And then I had my friend in front of me, and she had a bucket of mud, full of mud and a lot of worms. So we were going in a staircase, and then she just like bent to the back, and I was on her back. So that bucket just came to me. And then I was like, oh my gosh, like all this mud was very smelly and dirt, but I was fine. I just cleaned myself. But then later on, I just found this polychaete on my hair. So basically he was living on my hair for a little while.
It's always the hair and the head. I don't know if it'd be any better, but something tells me like if it were on my hand, I'd be like, oh, worm, look, I can see it. I can move it. But the hair.
At least they didn't came to my ear. That would be terrible. Just because I have a lot of hair.
Yeah, makes you scrub extra hard in the shower for the next couple months.
Yeah.
Thank you, Mariana. Alejandro, you are next. Will you please introduce yourself?
Hi, yes, I'm Alejandro. I'm also a scientist on Holly Bick's team. I'm a PhD student studying primarily bioinformatics, which is like really a mixture of computer science, biology, and statistics. Um, so I never really thought I'd be working with nematodes. That was like probably the last thing I ever thought I would be doing. My first time that I actually went doing field work was probably last year, or I'm sorry, not last year, but like a few years ago when we were still in California. We went down to an estuary to sample sediment there. And so we had to go and get the sediments. And so, you get into the mud. I didn't realize how thick the mud was going to be. And so I literally sank about like a foot in to like my knees, almost fell onto my face, but I managed to stay, upright yeah, I literally had to like grab my leg so I could actually like get unstuck from the mud. But it was like really nice experience, very beautiful there. We had the stingrays literally swimming all around us, so it was gorgeous.
I love that attitude. It marks you as like really a true scientist when you're like, oh, I almost fell on my face in the mud. It was beautiful. The best experience. I think you're doing exactly what you're meant to be doing, right? Thank you so much. Nick, you're next. Will you please introduce yourself?
Hi, my name is Nicholas Roberts. I am a scientific researcher at the University of Alabama. I'm a PhD candidate, and I'm super excited to be here. I use computers mostly to study the animals that live in between grains of sand. And a gooey story from my past is that the last time I was in Antarctica, they put all the mud in a box, and because I don't care about the big things and I care about the little things, you really have to dig around in that box of mud. Turns out, if you're really tired and you're covered in water, you can slip and fall. And so I ended up making mud angels that day, and the only way to really clean yourself off is to get hosed off by your PI at 5 a.m. So yeah, my boss was very happy about that one.
I laughed a little when you said you fell. I'm sorry, but then you said you made mud angels, and I was like, oh, I'm not a bad person. That's fine. Thank you so much, Nick. Victoria, your turn.
Hi, I'm Victoria Vandersalmen. I'm also a researcher. I'm from the University of Alaska Anchorage in Dr. Sarah Gherkin's lab. And so I study a tiny crustacean called a chamaecian. I've been working with these guys for close to 10 years now. And so I don't have a gooey story, but I have a crunchy story. And so for me, I had been working with chamaecians in a lab for about a year before I ever got to see one alive. And so I remember being out in Prince William Sound, Alaska, poking around in the mud when I saw my first chamaecian moving. And funny enough, they're known as comma shrimp, and I had never seen them move alive before. And so watching them float around in the water, they actually kind of flick the way that you would think a comma would move if it could. So I remember pointing to another guide that I was working with just this last year, and I'd been talking about chamaecians for years. And he's like, this is what's taking you to Antarctica? And it opened up a really cool conversation, like why something so tiny can be so important.
Thank you for sharing your crunchy story. We do not discriminate based on flavor of black stories. I really appreciate that you told us how a comma would move. Okay, so those are the research scientists. That's everyone who's in a hotel. Like I said, you are going to meet the ship folks in just a second, but first we want to tell you a little bit more about the science and about the specific type of worms that the Bick Lab studies. That's Holly, Mariana, and Alejandro, and some others that are here. So Mariana, will you please give us some visuals? Because I know we're all talking about super tiny stuff that if you just looked at it would look like little specks of dust for some of us. So let's see them close up. And you are still muted. Gotta love Zoom.
Now, yes. Now, can you hear me?
Yes, thank you.
Later. Okay, so let's talk about worms. So probably when you hear the word worms, so this is what comes to your mind, that squishy, gooey earthworm in your backyard, or maybe this pretty cool game that actually it's pretty cool. I play all the time with my kids. I always beat them. I don't know if I'm good enough or they're just being nice with me, but maybe these are the two things that comes to your mind when we talk worms. But today, what we want to hear is show you that is the entire universe of different worms living in the marine ecosystems in the oceans. So let's dig a little bit. Okay, so for example, if you go to the beach and you are playing around the sand and you get a handful of sand, what do you think we have right there? Probably you think that we have nothing, we have no life, we cannot see anything. So basically you see a handful of sand and no life at all. But what if I tell you that on this handful of sand, we have thousands of little creatures living there? So that's the case. So if you put this sand under the microscope, so you're gonna see that it's actually this sand, it's full with the little creatures. It's home to many extraordinary organisms. And all these organisms living together in between the grains of sand or mud, we call them meiofauna. And they are pretty cool. So you can find all these different shapes and colors. So you cannot think that we have all this on that, just a little bit of sand, but this is what we have when we see under the microscope. And here I want to show you very quick a video. It's just a compilation of a bunch of organisms that live inside of that handful of sand. So you can find all different kinds, shapes, colors as I see, and it's very cool to see these guys under the microscope moving. And for me, it's fascinating. Like just put a plate under the stereoscope and see that world that I talk about it. For me, it's an entire new universe. Probably we have some worms here that other people work with. Specifically, I work with the nematodes, but I really like the other ones as well. And some are very cute, I have to say. Okay, so now that you know, what is meiofauna, and I have to say that the meiofauna, even though they are very small, they are very abundant and diverse on the ecosystems. And what do I mean with abundant and diverse? So that is a bunch of these organisms in the sand or mud, and all kinds of organisms, very different. So specifically inside of the meiofauna, I work with a group that we call, actually they are awesome. For me, they are the best group on the meiofauna, are the nematodes. So they are very abundant and they are amazing. What I think it's very fascinating about them, it's when you see under the stereoscope, just looks like a regular worm, not much color, it moves really fast, and then you cannot see many interesting things about it. But then when you put in the microscope, you just see all these structures. So if you see this picture, maybe you can think oh my gosh, this came from an alien movie, or I just thought here that a good name for the nematodes is Stranger Things, those monsters, or the game, the Subnautica, all those sea monsters. So I always remind the nematodes, but we cannot tell that they have these structures until you see them under the microscope. So they have all these structures that looks like monsters, right? So these are my pretty cool nematodes. So they live in the meiofauna, and as I said, they are very diverse, very abundant. And as Victoria mentioned, that's amazing how such a tiny thing can lead you to different places. And as I said, I have been working with these guys has been 20 years. And what I think it's very awesome about them, that they are everywhere, so I can go everywhere to find them. I can dive, I can go to the beach, and I'm here in Antarctica just to find these tiny little creatures. And I'm very glad for that. So I hope you enjoyed my warm talk. I hope this was cool to see some nice creature living inside of the grains.
Thank you, Miriana. That was great. We are going to hand it over to the folks on the Nathaniel B. Palmer now. That's the ship in the upper left corner of this slideshow that you can see right there. They are going to take us on a ship tour. So take it away. Please say your names and let us know what we're seeing. Thank you.
All right, good morning, everyone. My name is Alex. I am one of the electronics technicians on the ship here. So I take care of all of the scientific gadgets that we throw over the side, everything that blinks or has little blinky lights on it, water sampling, cameras, all that kind of fun stuff. So I'll be showing you around the ship here. Matt, do you want to give a quick wave? Matt is one of our marine technicians on the ship. He takes care of a lot of the instrumentation that doesn't have electronics. He's behind the camera, so he doesn't have a mic on. So I am standing here on the back deck of the ship here in the bustling container port of Littleton, New Zealand. As you can see, sun has not risen yet. Nice and early. I have not had enough coffee yet. What we are looking at here, we have the A-frame behind us. So this is where we deploy most of our scientific instrumentation off of. Sometimes we can be sending out instrumentation several kilometers behind us. So we need a pretty big, strong structure for that. I say kilometers because I'm on a research vessel. I got to use the metric system. Sorry, guys. So also over here, you can see that we've got some stuff strapped down and ready for transit. So we're about to cross a pretty rough open stretch of ocean. So everything we have on board has to be bolted or chained down. Otherwise, it will get washed away. In fact, on the ship's transit over from New Zealand, we have a couple large shipping containers on deck that got pretty much damaged irreparably by waves smashing into them. So it's a pretty serious place out there. So we got to take it seriously. This whole deck back here could be a wash, could be waves up to my knees. So I'm going to take you on a quick little walk around the ship. Just don't have time for the whole ship, unfortunately, but just a quick taste of what life is like here. So as we come on through here, we're going to come into our first workshop. This is where the marine technicians, like Matt, who you met earlier, live and work. Well, mostly work. The marine technicians are sort of like our Swiss Army knives on board. They do a little bit of everything. They run small boats, they deploy instrumentation, they fix things, they break them, they fix them again, they fabricate equipment for us. So they really have to have a lot of tools at their disposal. So we have welding equipment, we have machine tooling, pretty much whatever happens out there, we need to be able to fix it and work on it. And so this is where they do all of that. So we're going to continue forward, leaving that shop, past all of our lovely safety equipment here. And we are coming into the aquarium. So on some cruises, we would use this as an aquarium for storing live animals, but this cruise, it's mostly storage. As I said, we've got to keep all of our sampling gear inside right now. If we're out on deck, it would get washed off. So what we have right here, this is the epibentic sled. So this will get a lot of use for sampling a lot of those little critters down in the mud. So we put this down at the end of a long trawl cable, and this tows kind of through that nice soft soupy mud with all the nematodes and funworms in it. And these bags back here can collect all that little stuff. Now we have other nets that we can use for the other researchers on board who are looking at, you know, the bigger things, the sea stars and all that, but this is really good for your little stuff, your myofauna, all those wee little critters. Now, the other thing that we'll be using a lot on this cruise, we'll be doing a bunch of coring work to collect the sediment and that sort of interface between the water and the sediment. So this is a tube off of our megacore. And there's a whole instrument that has 12 of these mounted on it. And that will drop down to the seafloor and drive this into the mud and then plug both ends. And so we can bring up a sample of that sediment. Pretty awesome stuff. But like I said, all that's stored up top right now. So I'm not sure if we'll be able to show you that. So it's all shiny and brand new. I'm really looking forward to getting that all muddy and scratched up. So continuing on. So this is our first lab. So you can kind of imagine this is the path that a sample might take as these critters come on board. They come off the deck after they get sorted, come into the hydro lab here. As you can see, still a little bit to unpack here, but this is kind of what life is like as we get ready for a cruise. All of our various scientific equipment spread out. Takes a lot to run an operation down here. So continuing forward. Next up, we have what we call the Baltic room. Now, the Baltic room is home to one of the pieces of equipment that I take care of, one of my best friends. So this here is our CTD. It is a water sampling equipment. So these big bottles on the side, I can actually trigger them remotely from the ship. We'll drop this down into the water. Could go down 2,000, 3,000, even 6,000 kilometers, 6,000 meters, sorry. 6,000 kilometers would be impressive. And in addition to collecting water from those depths, it can also take measurements on all kinds of different water chemistry as well. So pretty amazing piece of equipment. And one of the coolest things about it is when it deploys out of here, this whole side of the ship here actually opens up and swings open. And then the whole boom goes out and the CTD here drops down. And it dangles on this wee little bit of cable off that big winch there. We have 3 different oceanographic winches like this that deploy all of our equipment and are responsible for getting our science done. So there's a lot of really cool technical details on our side of things that make this science happen. So let's keep walking. Next up, as we continue forward, we're going to take you into some more lab spaces, give a little more of an idea of what the scientists will be doing. As we continue past here, this is another one of our labs. We call this one the dry lab because this is more getting away from the back deck, getting away from getting water everywhere, got more microscopes and things in here. As you can see, also a lot of equipment. Now, the other thing we have in here is a whole range of freezers and refrigerators and incubators. And several of these freezers are actually set to -80 degrees Celsius. So that is very, very cold. And we need those temperatures in order for the scientists to be able to properly preserve their specimens to bring them to home to study. So a lot of storage space on this particular cruise for that. We also have some larger freezers as we go over this way. One of them that we affectionately call Big Antarctica. It's our walk-in freezer. Camera, okay. Yeah, so we have a couple walk-ins on the ship. So that allows our scientists to do some in situ studies on things. They can actually kind of keep stuff at the temperatures they would be down at the seafloor, potentially around -1.8 Celsius, depending on where we're working. A few other lab spaces around the ship. The cool thing about the Palmer is that she's a really versatile workhorse. We have to be able to do all kinds of science. Last cruise, they were studying seismic stuff. They were doing geology. Now we're pivoting to work on invertebrate biology. So we really have to wear a lot of hats here on board and be able to kind of jump back and forth between different kinds of science. So she's got a lot of capability. So everyone wave to Carl. So we're going to step here into one of the most important parts of the boat, and that is the galley. This is where we eat. So we always get a lot of questions about what food is like on the ship. Food here is really great. The galley, big communal eating area. Yeah, it's pretty solid. The tricky thing is always after the end of a couple months at sea, fresh food starts to run out. So we always are looking forward to that first apple, first salad after a long research cruise. Other most important thing in here, we have our coffee machine. Some people may think that a ship runs on diesel. It does not. It runs on black coffee, as all good research vessels should. So the other bit of life on board, I'm going to take you upstairs and show you one of our cabins, so you have some idea of what our living spaces are like. All right, just a little bit further, a little further. Some of our friendly other techs holding the doors for you. So if we step in here, this is what one of our cabins looks like. It's not huge, but compared to some boats I've worked on, I would say it's pretty comfortable. We got 2 bunks here. We share our cabins on board. Given the number of people, it's a necessity, but realistically, pretty cozy. I've worked on plenty of boats where I didn't have enough space to stretch out, so it's pretty nice. All the cabins have a little ensuite bathroom here with a shower. Little taste of what it's like, you know, simple, but it works. I'm doing it time. All right. So we can have a quick glimpse up at the helo deck if we still have time, and then I will pass it off. So we're moving aft on the ship again, just to give you a little bit of orientation. It's easy to get lost on a big boat like this. So I just wanted to give you all a quick peek up here. So the Nathaniel B. Palmer can also, and has in the past, used a couple helicopters. But at this point, the helo hangar, which is here, mostly gets used as storage, because a trip like this uses a lot of equipment. Everything from cold weather gear for the scientists, extra spare parts, all manner of things. So this is this whole area. I alluded to earlier that most of our equipment is currently stored up top because we don't want it to get washed away. So this is where it lives, up here in the helo hangar, or back there on the helo deck. So I'm going to take a quick spin back there, and then I think that will conclude our little walk around to the ship. So we do have some fun things, little ping pong table, little disc golf thing, just to keep ourselves entertained on those long crossings. We've got a couple shipping containers up here. One of them houses a laboratory for working on radiation, and as well as spare parts over here. So I think we're coming to the edge of my connectivity. So I think that's about what I can do for my tour. So I think I'm ready to pass it back to you, Virginia.
Thank you so much, Alex and Matt. This was just like any reality TV show I'd see touring someone's house. I really appreciate it. I know that we don't technically need helicopters to study the deep sea, but I think there was still a part of me that was rooting when you went up to the helo deck that there was actually going to be a helicopter there, just because that's like Bond level science. So not for us this trip, but that's all right. I would like to take a minute to remind everyone we have at least 20 minutes, a bit over, just to answer your questions. Any questions? No question is too weird. We were doing something like this with adults the other day, and they asked us about pooping. So if you really have like some question like that, we're game. That's why we're here. So I'm going to start with Holly. I'm going to pitch this one to you. We have a question. How do worms and microbial stuff live in Antarctica in the dark, cold, deep?
Yeah, so fun fact, worms live literally everywhere. So you can walk outside your house or your school, wherever you are right now, and all you need to do is find a tree or find some dirt. And if you just scoop up a handful of dirt and take it back into the classroom or your home and put it under the microscope, you would see thousands of worms living there too. So nematodes are one of the most successful groups of animals on Earth. And by successful, I mean they are abundant and diverse pretty much everywhere, from the soils outside your house all the way down to the deep sea. There are different species depending on where you are. So the ones, living in the trees are pretty distantly related to the ones down in the deep sea in Antarctica. But they are just incredible because they can eat anything from bacteria to, microbial plants or plant roots. There's some nematodes that have like little spears for sucking out juices from plants. There are ones that eat other worms. So they're parasitic nematodes that eat other nematodes. The big ones eat the small ones. And they can live everywhere just because they have all these different biological strategies to be able to invade different habitats and survive. So the species differ depending on things like temperature and pressure and the amount of food. So in the deep sea, the worms tend to be like pretty tiny and small because there's a lot less food there. So they've, from an evolutionary perspective, they've adapted to deal with very low food inputs that are not reliable. So they kind of sit and wait, and then when food falls from the surface of the ocean, like all the plankton falls down, then they just snap it up and, you know, gobble it up, and then they can sit around until the next gob of plankton falls. So yeah, I mean, that's part of what my lab is studying. We don't really know why they are everywhere, and the diversity of the species is high. We always say there's anywhere from 1 million to 100 million nematode species, but we really don't know because it's difficult to study these tiny animals because they are so tiny. So we need to be specialists in terms of our ability to look under the microscope and define species. And that's what we're working on. So my lab also used DNA sequencing and genomics, and we're really trying to understand the worms a little bit better because there must be some big biological reason why they are everywhere. But they are, you know, think of nematodes and other worms just like insects. So the diversity is almost as big as bugs and beetles and things like that. There's just many, many fewer people studying the worms versus the beetles and things like that. So anything else? Did I answer that correctly? Yeah, no worries.
You answered it very thoroughly. I love
Okay.
any discussion of why a thing lives where it does that's primarily based on food. That's my kind of, I can relate to that. All right, we've got a lot of questions. Thank you so much out there, everybody, for submitting stuff. We're going to get to as many as we can. So we're going to try and keep these answers short. So if you have any follow-ups, make sure you put those in the chat as well. So I'm going to open with, how did you know this was something that you wanted to do? And then how did you arrive at like this career point? So I think Victoria, let's go to you first. How did you know, and then how did you get here?
Yeah, so when I was an undergraduate, I actually really wanted to study jellyfish. That was what I was excited to do. And I was looking for a scientist that I could learn about jellyfish from. And in that kind of hopes of finding someone, I met Dr. Sarah Gherkin when I was taking an oceanography class. And she's been working on the tiny little invertebrate called a comasian for years. And she had such a passion and excitement for them that I reached out and I asked if, you know, I could tour her lab. And truly learning from someone that was so excited about something, it was infectious. And so I just wanted to learn everything that I possibly could about them in the lab setting. And then I work as a tour guide in Alaska in the summer on a live-aboard ship. So I was able to take people to shore and have them look in the mud with me to look for comasians, and really seeing that excitement of people getting up 7 a.m. during low tide to look for comasians was really exciting to also see that I could get other people excited about something so small.
Fantastic. Anyone else in the hotel side of the research scientists want to say, how did you get this job that you ended up? How did you arrive at being a scientist right here?
Well, I can tell a little bit. Like, as I said, I have been working with this, has been 20 years. And interesting thing, I didn't want to work with the nematodes as well. I think like most of the people that I think about working with the ocean, I always thought like I want to be in the ocean. And interesting thing, like I always thought one of my biggest dreams would be go to Antarctica in a boat. I always said that like I want to be in a boat, like in a big cruise, sailing the ocean and studying the creatures in the ocean. But I always thought about the big ones, right? Like the ones that you can see, that they can blink. But I ended up in a lab that works with myofauna. So I started with all the bigger ones, like the macrofauna. But then I met the nematodes, those tiny little creatures. And this was how I got here. So I met Holly in my previous university, UCR in California. And I think because I know how to identify a nematode, basically, she got me right. And see, these tiny little creatures can bring me And that's the interesting thing, like that I said that I always wanted to be in a vessel, but then when I was working with the nematodes, I never thought that I could do that. Like, because at some point I said, like, okay, I'm not working with the big ones. Now I'm working with the small ones. It's going to be hard for me. But now I'm here in Antarctica and I'm going to find my little creatures.
That's lovely. Thank you so much, Mariana. Crew on the Palmer, we have a question. Have you ever seen anything weird out at sea? Y'all are muted, just so you know.
There we go. Try it out. Not necessarily weird on the last cruise, but on the last cruise, we actually had the joy of seeing the green flash. So right as the sun dips below the horizon on a smooth, calm day, you'll see a flash of green pop over the horizon, which is really unique and pretty rare to see unless you're far away from shore and in a very calm sea state.
Fantastic. I'm going to pitch it back to you and ask, what is the coolest animal you have ever seen while on board the ship?
Ooh, that's hard.
I mean, how much time do you have?
I'd say the current favorite animal has to be the emperor penguins. We were working with a group on the last cruise, tagging and looking and studying a group of emperor penguins in the Ross Sea. And it was very, very cool, very great to get a chance to be near those animals and just interact with them in their natural habitat.
Very cool.
Wildly range depending on what it is that we're studying on this cruise. It's probably going to be Cumasians that are going to be some of the coolest animals I've ever seen.
Yeah. Or some of the giant sea spiders, big enough to fit on your head.
With all the discussions about things in your head and your hair, I like, I mean, big, small, I don't mind, but like, my hair is my hair. Good gracious. All right, we have a question. What do you want to learn from this trip? Nick, I'm going to hand this one over to you.
Sure. This trip is great because not only do I get to learn a lot of things, but because so many scientists are in the same place, we get to learn from each other. Me personally, one of the things I want to learn from this trip is all of the undescribed and new species that live in the part of Antarctica that we're going to. It's really easy to see things when they're big, but when things live in between tiny, tiny grains of sand, sometimes you'll discover something that is totally new and you won't even realize it until many years later. So that is probably the thing I'm the most excited about, is finding new discoveries in the sand. And there's a lot of mud and sand at the bottom of the ocean. So I think we have our work cut out for us. So.
And then Nick, I think this one might be yours as well, or something you can answer. Are octopuses related to worms?
Wow, are octopuses related to worms? That is a wonderful question, because what is a worm? Well, a worm is basically something that is tiny and a little squiggle, right? That's a worm, right? So Mariana and Holly talked about nematode worms, right? Well, octopuses are in a group called mollusks, right? And octopuses and clams and snails, they're all related, but there is a type of mollusk that is very closely related to octopus that is a worm. It's called an aplycophoran. And unlike all the other worms we've discussed, they wear a suit of sort of armor that they put all over their body. If you think of like the chainmail of a knight, these little worms will put little chainmail all over their body to protect themselves in the sand. And yes, of course, because I'm talking about it, they live in between tiny little grains of sand. So.
Thank you so much. Okay, I'm going to give the next question to myself. It is, what do marine biologists do when there is a dangerous animal like a shark? So I sit very quietly, hold my breath, because I know that my bubbles, if I'm on scuba, will scare the sharks away. And I hope it comes really close so I can get a great look at it. There are so many things in the ocean that are dangerous if you make them dangerous, or if you're accidentally making them dangerous. So like, if you accidentally go too near something's nest, it may feel threatened. And then it says, I have to attack you to scare you away. but most of the time I've spent hours snorkeling and scubaing in the ocean. And in most of my hours, everything is so scared of me that I'm spending most of my time being like, wait, come back. So most of my work in the ocean has been spent not chasing them in like a mean way, but trying to figure out how I can get to them, to see what they're doing, to see what they're like, and to learn more for my own work. So I guess I would say back that I don't think of sharks as dangerous as much as I think of them as fascinating, as long as I'm being very careful and planning my work out very well. Okay. I'm going to give to Alejandro, how old were you when you were first inspired to be a marine scientist, and what inspired you?
Okay, I think I'm gonna be like the only person that did not think about being a marine scientist when they were a kid here. It's definitely not something like I think going to college, I always thought, okay, I'm gonna be a doctor when I first went in, and then that just did not happen because I started talking to a student, a medical student, and I was like, okay, I do not like that lifestyle. And so it just wasn't for me. And then I was just like hopping around majors trying to figure it out. And I literally was talking to a professor and I told her what I was interested in. And she told me to go talk that there was a new professor at the university who she thought I could really learn from. And she's like, you should go talk to Holly Bick. And so I literally went downstairs to her office and talked to her. And that's pretty much how I started on this on this like career path, is really just talking to Holly. And that's how I got introduced to nematodes.
I love that. And I just want to say for all the audience members out there that so many people on the ship, you're meeting just a small portion of us. There are, I think, 31 scientists who are coming on board. There's at least 20 crew. All of us had, some of us had kind of straight career paths where you go to school and then you're out of school and then you get a job. But a lot of us had really winding career paths. One of the scientists here did human resources work for a company for a lot of years. Y'all folks on the Palmer can talk about this too, if you'd like, but you can decide at any point, we think, that you want to make a change. And then if you really want to go after it, it's possible. We don't feel, I think, stuck in the kind of things we're doing most of the time. All right, let's see, Victoria, this one's going to go to you. If you weren't doing this job, what job would you do?
So if I wasn't a graduate student right now, I would be a middle school teacher. So after I finished my undergraduate career, I've been working as a tour guide and ship captain in Alaska now for 8 years. And in the off season, I would work as a teaching assistant at a middle school in Anchorage, Alaska. And I absolutely love that. But it was actually when Dr. Sarah Gherkin, who again was my undergraduate research advisor, she approached me and asked if I wanted to go back to studying chamaecyans and if I wanted to go to Antarctica with her. And how do you say no to that? So here I am. But I always think fondly on when I was a teacher, and it was really a lot of fun.
Thank you, Victoria. Folks on the Palmer, I have one for you. Have you ever been in danger while working on this kind of stuff?
Yeah, absolutely. So a number of years back, I was working with a group studying elephant seals and Weddell seals. And in order to work around these animals, you are in a relatively close proximity to them, and the concern is the safety of the animal and the safety of the scientists. And once we approach the animal and begin the tagging process, we need to make sure we're taking care of that animal the whole time. So we're kind of in a close proximity to them. So while nothing happened in those particular scenarios, we were just close to these animals, and that's a dangerous position to be in. So you need to make sure that you are aware of your position around with your teammates. So we work as a team in this entire process. Making sure that both we are safe, the boats around us are safe, and the animal itself is safe. So those are kind of the dangerous scenarios you could be in. But we take a lot of time to plan and work together as a team to make sure that they all go off smoothly. And that was another very successful cruise. We had over 30 animals worked on during that project, and it was extremely successful for the science teams. So we're hoping to again do that in the future.
Thank you so much. I would echo that teamwork makes everything better when we're doing this kind of work. We have about 5 minutes left for questions, so I'm going to just, again, keep going as much as I can. I love these questions coming in from y'all in the audience. Thank you so much. Holly, you are the person who has like done the science thing the longest out of all of us. Somebody says they are wondering how much a researcher makes a year. Can you walk us through the salaries?
Yeah, we're definitely not in this for the money. So that's what I will say. We're in this for the joy of the science. Yeah, it varies. It varies. But to give you some specific numbers, typically graduate students make about $30,000 a year, which is not a lot of money. It's a fairly low paid training position, but you have the potential to make as much as you want really after grad school. So if you were to do an academic career path like I did, so if you wanted to be faculty at a university, typically you would do a postdoc after grad school. Nowadays, postdocs make between $50,000 and $60,000 a year. And then faculty, once the career stage after after a postdoc would be faculty. So you start as assistant professor if you're going to be a tenure track faculty. In universities, the salary range is 70,000 to maybe like 90,000, depending if it's a state school or a private university. And then, once you get tenure, your salary is usually over six figures. Probably not high in the six figures, but $100,000, $150,000 to $200,000 for a full professor is not unheard of. So, I'm totally happy with that salary. It's enough to live on. Someone like Alejandro, who's maybe not going to go the academic career route, if he wanted to finish his bioinformatics degree and go work for Google or IBM, he could totally do that. And he would make way more money than me, like immediately after grad school. So I have friends who work in industry that make easily starting salaries of $150,000, $200,000, $250,000 a year as data scientists working for private companies. So really depends what you want to do. And especially if you're on the genomics and data science side, biotechnology, the salaries can be really, really high in the long term.
Thank you, Holly. All right, I'm going to try and squeeze in 3 more questions real quick. Mariana, how do you tell if a worm is a boy or a girl, and how they make baby worms?
Yes, you can see if they are boys or girls. So the boys, they're gonna have something that you call a spicule. So that's like the boys' identity. And the female, they have also an opening, so we can tell that they are female. So basically what the male worm does, he's gonna look for the female, he's gonna find the female, and then imagine that this is the worm, like the female, the male just goes around, wrap that female, and then they make babies. And then it's a lot of babies, just like this, little eggs coming from outside of the female. That's something amazing to see under the stereoscope. You can see the eggs. So that egg is going to be inside of the female for a little bit, and then they're just going to release the babies, the baby nematodes, and they're very cute.
I'm giggling a little bit at you just watching this whole thing happen. It feels a little invasive as a person.
It's fun. It's fun to see like the babies coming out of the female.
Love it. All right, 2 more questions. With so many scientists, Nick, this one's for you. With so many scientists, how do you divide up lab time and equipment usage?
Wow, that is a really good question. So when you have 30 plus scientists and 20 plus crew, right, everyone's going to be wanting to be working in the lab. You're in Antarctica, right? It is so exciting. And so what they do is the boat will, once it starts working in Antarctica, work 24 hours around the clock. So as you're going to bed, your fellow scientists are waking up and taking your spot at the microscope. Dividing up equipment and lab usage, the scientists have a lot of respect for the marine lab technicians and marine technicians. So when they say stuff's going down and stuff's coming up, we listen to them and follow what they have to say because they know the safety the best. And we'll take whatever mud we got and bring it in. And then if it's bedtime, I'll knock on the door of my buddy and he'll wake up and take my spot at the microscope. So 12 a.m. to 12 p.m. and 12 p.m. to 12 a.m. So
I'm looking forward to claiming the day spot as much as I can get, so I can eat my meals when meals are supposed to be eaten. But I would wake up for something exciting. Let's just say that too. And then I think the last question we have time for, Alejandro, is it fun to be a scientist?
Absolutely. I think, you know, when you make a discovery and like, for example, you know, this is the first time we're sampling this area of Antarctica in like a couple decades. And so knowing that we're going to have these pretty like new samples, and then we're going to look through it, make these discoveries. And at that moment, we're going to be like the only people who know about this. Like, that's just like amazing and mind blowing to think about. But I would not be here if I did not enjoy this. And also being able to, you know, go to these remarkable places like Antarctica.
Thank you so much. And thank you to all our panelists. That is unfortunately all the time that we have. I'm going to put some links into the chat, or I think you will receive them by email after this is done. We have lots of ways you can stay in touch with us. I think Sarah is going to tell you more about that maybe right now.
Yeah, absolutely. Thank you all so much to all of our researchers, our staff on the Palmer. This was absolutely fantastic. For anyone who's interested in learning more, and as Virginia said, I'll be sending out an email in a few days with all of these links. You can also use the QR code we have here on the screen. You can find out more about Holly's research and Virginia's work, as well as the U.S. Antarctic program, which is operated by the National Science Foundation. We've got photos, videos, we have a ton of educational resources as well. And obviously you can follow all of us on social media, and I'll make sure that Holly's research team, their social media is also noted in my follow-up email. I'm going to leave this slide up for a few minutes so that folks can jot things down if they like. But if I didn't say this, I would be remiss to thank you. Thank you all so much for your time. I know it's very early where you are. So I guess go take a nap. And to all of our attendees, thank you so much for joining, and keep an eye out because we'll continue to do live events from all over the polar regions. All right.